this series, we talked to the scientists at the Dangoor Center for Personalized Medicine to get a closer look at what's happening in their specific research laboratories, what developments are expected to reach the market, and what they think medicine will look like in the coming years. This time, we're lucky to host Professor Nissan Yissachar. Professor Nissan Yissachar is from Bar-Ilan's Faculty of Life Sciences and the Institute for Nanotechnology. His research focuses on intestinal bacteria and their connection to the immune system and various diseases. He holds a bachelor's degree in biology and a master's and doctoral degrees in molecular biology from Bar-Ilan University. He also served as a postdoctoral fellow at the Weizmann Institute and then at Harvard University in the United States. Currently, he heads a leading laboratory in the Faculty of Life Sciences at Bar-Ilan. Welcome.
Thank you
for joining us.
Sharon, thanks for having me.
So let's jump in and start with a definition.
Okay.
What are the gut microbiota?
Okay, so the gut microbiota are the microorganisms, basically a collection of trillions of microorganisms, mostly bacteria, but also viruses, fungi, and parasites and others. And these microorganisms populate our digestive tract, our gut, but basically every other surface in our body that is exposed to the environment, like the skin or the urogenital tract. Most of these bacteria reside in the gut, and we know that for a while, but only over the past decade or so, we understand that these microbes have a crucial role in affecting our physiology and health.
How did we figure that out? Yes, I know we've known about, we didn't call it the microbiome, but we knew about the fact that our bodies held all these bacteria inside of them. We also knew that if they got out from there, it could cause problems. But what changed in the last decade or so that essentially led to this explosion in understanding the relationship between the microbiome and the rest of our body and mental health, I've even heard?
Exactly. So, up until recently, we could not study bacteria if we could not grow them or culture them in the lab. What actually changed over the past, let's say, 15 years is advancement in the DNA sequencing and then computational methods, which now allows us to characterize and ask, okay, which bacteria reside in my gut, in your gut, without the need to culture them in the lab. We just collect, for example, for gut microbes, we usually collect the stool samples, we extract the bacterial DNA, and then sequence their DNA, and that gives us a kind of a long list of which bacteria reside in my gut and in which quantities.
Do you and I have the same gut microbiome?
So apparently these are like a kind of a bacterial fingerprint. Each one has its own unique bacterial ecosystem. And we're talking about very, very large number of bacteria, trillions of them.
Trillions.
Yeah, and the number of bacterial cells that reside in our body is equal to the number of human cells that make up our body. So we are kind of a superorganism that is composed of both human and bacterial cells.
That's a little creepy. So you're saying like
Crazy.
half of me
half
is
of
not
you
human.
Exactly. And actually, if we count the bacterial genes that make up your body, we're talking about over 99% of your genes, your body's genes, is actually microbial genes. So we are 99% bacteria by any scientific standard, basically.
Oh, well, that's a little disconcerting. I'm going to put that aside for a moment. We're going to go back to the gut microbiome.
Right.
And I'm going to think about them living in this contained space, and the rest of me can be human. But so you're saying that we've learned, because we're able to extract them or use samples, fecal samples, that we could go in and test what everybody has. Are there good bacteria or viruses and bad bacteria and viruses?
Right. So we used to think of bacteria as bad creatures that makes us sick.
But antibiotics. We kill them.
And antibiotics, let's kill all the bacteria.
Right.
Honestly, the vast majority of the bacteria are not bad, are not pathogenic. They don't cause disease. They colonize our body. They reside in our gut. And it's very good that they are there because they are not just passengers. They are actively shaping our health, our immune system, our nervous system, even our behavior sometimes. So it's considered to be very. It is actually a very positive thing to have a complex and rich ecosystem of bacteria in our body. Okay.
So let's dig a little deeper in that, because it isn't intuitive that something that lives in our gut can impact the rest of our body, right?
Right. This is that was really mind-blowing in the past 10, 15 years to realize that things that happens in the gut, the gut is not Vegas. What happens in Vegas stays in Vegas. What happens in the gut doesn't stays in the gut.
We're talking about the Vegas nerve.
Right.
Okay, wait, not everybody, I got it. But let's just, you want to explain the difference between the gut and the vagus nerve a little bit so that everybody can understand the joke.
So what happens in the gut affects throughout the body. Bacteria in our gut can signal to the brain, for example, by nerves that connects the brain and the gut, and by that affecting our behavior, or even neurodegenerative diseases like Alzheimer and Parkinson. These bacteria also educate our immune system. So cells of our immune system that reside in the gut, get signals and become mature or active based on signals from our gut bacteria, and then these cells can traffic from the gut to the rest of the body. And then initiate, start inflammation, stop inflammation, and that really, regulate health and disease. It's a very important factor in autoimmune diseases like multiple sclerosis in the brain, or diabetes in the pancreas, or arthritis in the joints.
Just to make sure I get this. So there are bacteria that live within our gut, right? Within, our digestive tract, essentially. And I am sure they live in different parts of the digestive tract, right? More some than others. And you're welcome to dive into that a little bit more. And then they are sent out through what mechanism? How do they get to our brain? They cross the blood-brain barrier? How does this work?
So in most cases, we actually don't know. That's what we're trying to figure out in the lab.
Oh, good. So I don't feel bad that I don't know either. I'm in good company.
Exactly. But we do have few examples. For example, bacteria can produce chemicals like neurotransmitters or other chemicals that affect our brain, our immune system, and other organs in our body. And they can also, activate cells of immune system that reside in the gut and then traffic and migrate to other parts of the body. Yes.
That's where you want to go. And the vagus nerve that you referenced with your joke?
Yeah, so this is kind of a cable that connects the gut to the brain and transmit information back and forth. And that allows the brain to sense what's going on in the gut, and allows the gut bacteria to educate the brain and affect brain's function.
Does it work the other way? Meaning, because I know when I get nervous, sometimes my stomach hurts me a little bit, right?
Right. And this is actually very interesting, because when we try to figure out how do bacteria affect our immune system, we found that one of the first responders that sense our gut bacteria is actually the nervous system. And besides the central nervous system, our brain, we also have a second brain, this kind of a smaller brain, which is the brain in our gut. And this is the enteric nervous system. Apparently, we have millions of neurons, brain cells, basically, that reside in our gut, and they sense the gut microbiome, they respond to the gut microbiome, and they target the immune system. They signal to the brain, and they also target the immune system to initiate inflammation, to stop inflammation, or to act in this way or another. And this way, it actually regulates, like our second brain regulates our health in the gut and beyond.
Interesting. Inflammation is a big buzzword now, right? I'm used to inflammation. I bang my leg, it swells up, it's inflamed, right? But it means something more than that, doesn't it?
So if we think of a cancer patient, for example, that receive immunotherapy, these drugs activate our immune system in order to fight, or naturally fight, tumors, cancer cells. So with this respect, inflammation is good, because we want to activate the immune system, activate the inflammatory responses to fight cancer. And we know now that most patients do not respond to immunotherapy. And over the past 5-7 years, we realized that some of the gut microbes that reside in our gut will determine if a patient will respond or will not respond to a specific immunotherapy.
Do you understand the process by which that works? I don't need to get too geeky. I'm fascinated by this.
So we have a couple of examples of how does it work. They basically, like in a simple way, they activate the immune system that migrates from the gut to the tumor, and then basically kill the tumor cells. So now in my lab, we try to figure out how it works and what makes one patient to respond and the other not to respond. And that actually brings me to kind of a unique method that we use in the lab. Maybe I'll start from the beginning. One of the most studies about gut microbiome focus on characterizing bacterial composition. Let's say we take fecal sample, we sequence that, and then we get a long list of bacteria and their quantities. Okay, so that's like having a grocery list. Okay, but we have the ingredient list. We have no idea what the recipe looks like. Basically, we don't have no idea what do these bacteria do. How do they affect the gut, the immune system, the nervous system? So what we're doing in the lab is that we developed a kind of a nice, a novel system that enables us to take small pieces of the gut from mice and to keep them alive outside the mouse in a kind of a mini gut organ culture setup that we developed, and then to introduce microbiota from patients and to ask, okay, so how do specific microbial configurations from that patient or these patients, how do these affect the gut, the immune system, the nervous system? And can we identify the relevant bacteria, the good bacteria that can be used to treat disease? If it's, in the context of cancer, it could be, which bacteria will activate the immune system to fight cancer. In other cases, like autoimmune diseases or inflammatory bowel diseases like Crohn's and colitis, we want actually to identify the bacteria that will shut down or ameliorate inflammation. So basically, the long-term goal of these studies is to identify which bacterial components can turn on the immune system or turn off the immune system in order to treat autoimmunities, inflammatory bowel diseases, and cancer.
Right, as you were saying, inflammation can be good.
Exactly.
Right, in the layman's term, or it could be bad.
Right.
In
exactly.
the case of autoimmune diseases,
exactly.
It's essentially your immune system acting in ways that are unhelpful. Whereas if you're fighting a tumor, you want it to attack the tumor,
exactly.
and you want it to be inflamed, if you will, right?
Exactly. So we can think of next generation therapies as one of the goals in our lab. Is to design new microbiome-based therapies. If we can identify which bacteria are relevant, which bacteria will be beneficial for a specific disease, how do they work, which product they produce, we might be able to use these substances or these bacteria as a therapy, either as a supplementation, as an adjuvant to immunotherapy in cancer or other drugs, or as microbial-based therapies themselves. So instead of getting chemicals to, for example, suppress your immune system in Crohn's, you might be able to get a bacterial cocktail as your anti-inflammatory therapy.
Although when you say cocktail, you're not really talking. I mean, where does the cocktail come from? Meaning, let's talk bottom line. From what I've heard from some of the labs at Bar Ilan also, that actually quite promising is this idea of fecal transplants.
Right.
So on the surface, it's a little distasteful.
Right.
No pun intended. Obviously, you put it into capsules. But talk a little bit about that treatment and its success, whether it actually has been proven to make a difference.
Yes, definitely. So fecal transplant it's kind of an old and new therapy, old because it was used, thousands of years ago by Chinese, in Chinese medicine. And even today in veterinary medicine, they can take fecal samples from healthy animal and transfer it to an animal with an infection to treat disease. In humans, it's relatively new in the kind of modern medicine, but over the past decade, it's even FDA approved for some cases of infections. One of the only FDA approved use for fecal transplantation is for an infection called C. difficile infection, Clostridium difficile infection, and that affects hundreds of thousands of individuals, mostly elderly, every year. Very nasty infection, and very often this bacteria is antibiotic resistant. So it's a life-threatening infection that in some cases we don't have a lot to do with that. And the very simple solution is fecal transplantation. If we know that the microbiome is good, not only to, the microbiome could be beneficial to educate our immune system and so on, a healthy microbiome could help us fight infection. So let's take a healthy microbiome from a healthy individual and transfer it to this patient with this infection. And in the case of this Clostridium difficile infection, it works like magic. Over 90% of patients, gets better, go home from the hospital the day after. It's amazing.
That's incredible.
Yes. So that really works like magic.
No side effects?
No side effects, almost no, very, very negligible side effects, if any, were reported. But this is for that specific infection. So obviously that was, mind-blowing. And so there are hundreds of clinical trials trying to take the same approach to treat almost any other disease you can imagine, from cancer to inflammatory bowel disease to autoimmunity. Apparently it doesn't work so well in more complex diseases.
Do you know why?
Cases. Yeah, because it seems that in most, in complex multifactorial diseases, we probably need to figure out what's the mechanism. How does it work? Which bacteria are actually the good ones, the necessary ones? Because, in fecal transplant, we just take, random bacteria from a random patient and transfer it to a random, from a random donor to a random patient. When it works, but we have no idea how and,
which specific bacteria are doing the trick, if you will.
Exactly.
Or even, I would assume sometimes bacteria work with each other. You need, because you said cocktails, you need
exactly.
combinations. This bacteria helps that bacteria, and this one will suppress it. And It's very complicated.
Exactly. So we now realize that in most cases we need to understand the mechanism. We need to know which bacteria will produce which substances that will be beneficial or required to treat which disease in which patient. So this is kind of we're talking about personalized medicine and so on. So this is the personalized medicine at its best. We expect that treatments will be tailored not only based on, for example, DNA sequence or genes of the patient, but also on microbiome composition. We can, we're doing blood tests, for example, so we can take stool sample, get a signature or description of your microbiome, what your microbiome looks like, and then we'll tailor the therapy according to that.
It would seem to me also eventually, I don't know, but if you could do that and it's not outrageously expensive, if you, which sounds like you're doing this work in your lab, if you know certain bacteria are good as prevention, right? People who have these bacteria are less likely to have dementia. People who have this bacteria are less likely to have an issue, autoimmune issue.
Right.
Then we could try to figure out how to populate our microbiomes that way.
Right. So it's still very difficult to determine which microbiome is good or bad because it's very complex by itself, but also in the context of individuals. So each one, with different genetics, different health status. So it's pretty difficult to determine which microbiome configuration will be good for me or for you. But I think that's one. We're going to get there in the very near future. What we already can do is to identify specific microbes that might be beneficial in some diseases and use them as either diagnostic, to say, for example, we can say, okay, this patient will respond to that therapy or not, or actually as a therapy by
So you're
itself.
actually there. You went from mice to human.
Yes. So in some cases, for example, many other labs in the world and of course in Israel, trying to do just that. The advantages or the uniqueness of our lab is this mini gut on a dish, that system that we developed.
Yeah, you mentioned, I went, whoa.
Exactly.
Mini gut on a dish.
So that allows us to test, individual microbiome from actual patients and to ask what they do. Going beyond this list of bacteria, it's like, comparing, if you have a list of guests in a party, okay, so which guests are there, but using our system, we can now say, okay, which one is talking, which one is, is making music or arguing or whatever. We can actually see the function of these microbes in live, in real time.
And it maps well from mouse to human?
In many cases, actually, yes. We had the pleasure of working with several groups in Germany, for example, in the context of autoimmune inflammation in the brain, multiple sclerosis. And we showed that we can isolate specific compounds produced by bacteria and use these compounds to treat disease in multiple sclerosis patients. And that actually ameliorates disease in like 90% of the patients.
Wow. So do you have a lot of collaborations?
Yeah, actually a lot. And Israel, actually, in general, we have several very, very, very strong research groups in universities, hospitals, biotech companies, and so on. And the ecosystem in Israel is very unique, and it allows us to transfer from the patient to the lab and back to the patient relatively easily and rapidly.
And internationally, you mentioned Germany. You have collaborations.
Right. So several groups in Germany and Munster University in Hanover and others. Of course, in the U.S., Harvard, Cornell, and other universities, and other places in Europe as well, like Italy, Spain, and several other countries.
Well, you were at Harvard, so you have connections there,
right?
So let's talk about you a little bit. Now that we're impressed with what you're accomplishing, how did you end up where you are today? How did you end up coming back to Israel? Let's go back even further. When you were a kid, what did you see signs of this? Did your parents say, he's going to research the microbiome?
No.
You're out there playing football.
No, I was playing football. I was, building like mini gliders and stuff, kind of reading books, a nerd, basically.
I was one also. So from one nerd to the other.
And then, in the army, I worked with, in the computer. I was an officer, team leader in the computers division. So I was, managing projects and in the computers field. And I also worked in the high-tech industry after I finished my army service. And then, I didn't even think about science until my girlfriend back then, but my wife now, she was going to study biology. So I said, okay, sounds interesting. I'm coming with you. So I signed up for Bar Ilan.
This was before you were married. That was
Yes,
a big leap of faith there, my friend. A lot of pressure.
Exactly. No, but it was really fun to study together. We did the undergraduate together at Bar Ilan.
That's
and we graduated. My wife also continued to PhD studies in Shiba Tel HaShomer Hospital in collaboration with Bar Ilan. And she later on worked in Dana-Farber. And now she works at Schneider Children's Hospital. So we did the whole track together in different labs. And then, but thanks to her, actually, I did my undergraduate biology. Looks interesting, so I continued to master degree, which was interesting. So I continued the PhD, and then a postdoc, and a second postdoc, and I'm in biology ever since.
So the short answer is you did it for love.
Exactly.
We get the
you have
benefit.
to love what you do, yeah.
Well, you have to love what you do, and clearly the person you were doing it with.
Exactly.
That's part of the story here also. So we owe your wife a big thank you for getting you to be this incredible researcher. And why Bar Ilan? Sounds like you're deeply connected to Bar Ilan.
Right. So actually, love is the same answer here as well, because we have several opportunities, both for undergraduate studies or graduate, and also when we return from the US back to Israel. Several opportunities in different universities. I just love Bar Ilan, basically. I love the faculty. I love the diversity in the university. It's very diverse, people from all, kind of a microcosmos of the Israel society. The interactions in the faculty, in the university, between faculty members, between students, it's very friendly. It's very kind of family-like interaction. So I love that. I love coming to work and, just, having... fun at the lab, but also having kind of family-like environment.
Sounds wonderful. You're in the Institute of Nanotechnology, which is also
Right.
this very unique space, right?
Right. It's a combined space. We have labs in each and every floor has labs from different disciplines, from chemistry, biology, physics, and that nurture a lot of collaborations and interdisciplinary studies that are really crucial to do, cutting edge science.
Right. So it's not enough to study one thing. You need to be able to integrate in technologies and different fields. And so now you have colleagues next door. You could drink coffee together.
Exactly.
Right?
We do.
Yeah, very different conversations around your coffee than in other places. So you're not talking about the, Sidrach, the television episode from last night quite the same way. Yeah. Or maybe a little bit also.
Usually both.
Okay, very good. It sounds like a healthy balance there. So if I were to ask you, okay, you've got a magic wand, where do you want to go in the future? Give me your vision, and then we'll talk bottom line where you think we're really going to be.
Okay, so maybe the long-term vision is to transform medicine in a way that we can now use microbes as live therapeutics, replace the kind of the synthetic drugs or molecules that are used for most diseases now, and replace them with, active microbiome-based, bacterial-based therapies for inflammatory bowel disease, autoimmune diseases, cancer, and sometimes maybe even, like brain disorders, neurodegenerative diseases, behavioral disorders, and others. We already have some very promising results, very promising candidates of microbes that can modulate our immune system and our nervous system, which we now test in the lab in mice, and hopefully to transfer them into patients. So the long-term goal would be really to identify these microbes, the mechanism they work, and to use them as therapies.
Why? Why is that so important to do?
Because I think that, it's kind of, first of all, personally, it's kind of I like the most, the ambitious, the aspiration is to use our knowledge, to use our research, not only as isolates, kind of not only to expand knowledge as basic research, which is also important, but also to use it to give back to society, to human health, to global health, and that's really one of the things that drives me every morning. The idea that we might be able to save lives, to treat patients, to improve human lives. So that's really, to make a difference.
So you answered the question completely differently than I was asking, but it was beautiful, and I'm glad you went there.
But fantastic answer to a different question.
It was my question, and you actually answered it in a way that I didn't mean, but it's actually really powerful and meaningful and valuable for us to know. What I meant was, why the microbiome instead of other medications? Is it an issue of side effects? What is it about the microbiome that excites you as a potential clinical solution?
Right. So, I mean, basically everything is important. Classic medications are important. Genetics and molecular biology are very important. For example, genetic editing technology like CRISPR and others are super important. So everything is important. But the truth is that what's one of the most unique features of the microbiome is that it is very easy to manipulate. For example, if we look at our own genomes, let's say we discovered a new gene that is now makes you sick. Fantastic. What do we do with that?
Right.
Currently, nothing. Maybe, hopefully, in the future, we will be able to use gene editing and to treat genetic diseases, RNA editing, DNA editing. Okay,
right.
But currently, nothing. Bacteria, on the other hand, are very easy to manipulate, to modulate. We can, if we would just know, which bacteria will be the right one to use, and we already have some preliminary clues and evidence, so we can just, add bacteria, subtract bacteria. Very, very easy to manipulate. And that, basically opens up a whole new field in medicine. Diseases that we thought to be untreatable, systemic, multifactorial diseases, genetic basis. We can treat them now with simply just by modulating our gut bacteria. So that's crazy.
It is. It's crazy. No, I mean, it's not crazy, but it's fantastic.
It's fantastic.
Let's say.
Yes, exactly.
And again, as I started, I said, I read about the microbiome, and the fact that it has such a dramatic impact on so many things. It also feels a little less invasive in some ways to me than putting in a chemical substance into our body. If we already have bacteria there, to just introduce other ones,
right?
Feels almost natural, as unnatural as you're telling me the majority of me is not human cell; it's bacteria. It feels unnatural, but it is natural. Right?
It is. Yes, we just need to gain more comprehensive understanding of what is a good microbiome for me, for you. How can we treat that? How can we transfer? Let's say if a person has an imbalanced microbiome, how can we transfer it to a balanced one, to a microbiome that will maintain your health, that will educate your immune system, or into a good way. So this will be, and I think that it's not too far in the future. I'm pretty sure that very soon this will be integrated to clinical practice. We already have now these days hundreds of clinical trials trying to do just that.
Right, but as you said, it's complicated, right? Because you can work on, in general, how a gut in a lab responds to something, but that doesn't mean my gut's gonna respond that way, right?
So we found out that most people are not mice. So it's very surprising, very striking. But of course, many findings do not translate well from mice or cells in a petri dish to the actual patients, but some do.
Yeah.
And this is the way science works.
But even more than even if you were to do it on humans, right? This is what personalized medicine is.
Exactly.
So
we want to know. Yeah, we can use microbiome as a diagnostic tool. For example, if you go to get a therapy for to cancer or to any other disease, we will be able to tell you, okay, let's take a stool sample and tell you, will this specific therapy will be useful? Will you respond to that therapy or not? But also as a therapeutic tool, we can give patients, we can supplement patients with the microbial cocktails that are personalized to specific disease and specific patients.
I love the word cocktail. We won't say fecal transplant. Okay. A cocktail makes me think of parties and sweet tasting things. But yes, I know you mean it in a medical sense, and it's wonderful. But you sort of answered the second part of the question when I said to you, give me your fantasy and then give me the reality. And you said you don't think it's that far off.
It's not far off. I mean, we already have in my lab, but in several other labs in Israel as well, very promising preliminary results. Hopefully we will be able to translate them to human patients very soon.
Is Israel at the forefront of this?
Definitely. We have a great panel of scientists and physicians that got very well, were very well trained in the top institutes, top universities throughout the world, and established new research groups in Israel, in almost any university, at the Weizmann Institute, Tel Aviv University, Hebrew University, Bar Ilan, of course. So pretty much any university in Israel has microbiome researchers now, and are really leading the field worldwide.
And of course, Bar Ilan is at the forefront. Of
course, Bar Ilan.
With your lab at the front there.
Actually, we have only in our faculty, the life science faculty, we have at least 4 or 5 research groups that actively do microbiome research. And if I look at Bar-Ilan in general,
the Faculty of Medicine has
the Faculty
more.
of Medicine, the Faculty of Engineering, Physics, Mathematics. It's
mathematics.
Even mathematics, because understanding these complex interactions required tools, mathematical models, data analysis, network analysis. So this is, and we have to combine all of these.
Do you work with that? Use AI?
Yes. Even in my own group, we are mostly experimental biologists, but we also have computational biologists in my lab that they do, data analysis, mathematical models, and network analysis, AI, and others. But also, in different faculties, in different departments. This field is really, really interdisciplinary field. We have to combine microbiology and immunology and neurobiology and computer science and physics and network science and AI and
everything.
Yes, that's what makes it so interesting and fascinating.
You can see how much you just even enjoy talking about it, which is great. So, it's a continuing love story, right? Started with a woman, which, behind every great man, they say, right? So behind every great man's success.
My wife says that behind every successful woman, there's a pile of laundry. I don't know if you will be able to use that in the podcast, but never mind.
I think they should keep it in there.
Okay.
So your wife speaks wisdom, right? So, and I'm sure she's brilliant in her career also as a researcher. So not just, brilliant, but wise, let's say, in everything. But it's been a real pleasure, really, to meet you and to see the kind of joy you bring to your work and the success you're having. I mean, hopefully I'll never need it and nobody I love will, but I suspect we will at some point. And even if there aren't any real diseases, it sounds like some of the things you're working on will be preventive and will make all of humanity, as you said with your first answer, you just want to make humans live better, healthier lives. Exactly. And so I appreciate everything you're doing.
Thank you so much.
And this has been fun. It's been great to have you here and learn about what you're doing and learn a little bit about you.
Fantastic. Thank you.
Thank you for coming.